Hello, and welcome to the Sleep Science Podcast. I'm Penny Lewis, a sleep scientist and the presenter of this show. In this program, we talk about all things related to sleep, from dreaming and sleepwalking to what sleep does for the brain and the body, and how we can get more out of our sleep. In this episode, I'll be talking to Vlad Vyazovskiy, an associate professor of neuroscience at Oxford University. Vlad takes a cross-disciplinary approach to sleep with the primary goal of understanding what it actually is. We talk about sleep in rats, mice, hamsters, seals, and plants, to name a few. Vlad will explain links between torpor and sleep, as well as talking about local sleep and the idea that small areas of the brain may fall asleep while an animal is actively performing a boring task. So, first of all, welcome to the Sleep Science Podcast, and thank you very much for agreeing to do this and spend your time. Vlad is an extremely prolific and interesting sleep researcher who really focuses on trying to characterize what sleep actually is, which is quite a different angle than most of the other guests we've had. And he also focuses on rodents and primarily actually on mice. So I wondered if I could just start by asking you to tell me a bit about sleep in mice and how it's special and how it's different from humans and other animals.
Okay, I have to correct you, though, that I'm working not on mice. My very first paper that I was involved was on Hungarian hamsters, and this was at the University of Zurich, where I joined as a PhD student with René Tobler and Alex Borbey, and they were very interested in a state called torpor. So it is a state of depressed metabolism, and we are still trying to understand how it relates to sleep. And I was helping with hamsters, so my very first paper is on hamsters. And then for a very long time, I worked on rats and mice, but now I'm involved in various projects on other species. So I was a co-author on a paper about slow lorises. These are nocturnal primates. I'm involved in a collaborative process on sleep in bats. We can talk about this separately because bats are a very curious example. I'm also co-supervising a student who works on flies, and I have hamsters again.
Okay, so maybe then a good thing to ask you would be, since you're working on sleep in so many different diverse species, can you talk a little bit about how sleep differs across species? What are the similarities? What draws you to all these different species?
All animals sleep, as far as we know. To be precise, relatively few species were studied systematically and carefully with respect to sleep. And most species were studied in very standardized conditions, 12-12 light-dark cycle, and a lot of food on the ground. And then you're sleeping right next to that. Very different from what you experience in the wild. Think about sleep, how it evolved. Obviously, it evolved in conditions which are radically different from those where we study sleep now. I'm talking really not only about mice, but also about humans. So this is why I think it is very important to step back and start studying sleep in different species, because animals sleep in very different ways. Sleep is exquisitely sensitive to environmental conditions. So we need to kind of embrace this ecological approach and also in humans, in human research.
And do you feel like some of the studies that you've done have elucidated aspects of how sleep actually evolved, how it differs between species?
Well, I cannot say this about my own research. In terms of phylogeny of sleep, I'm the beginner. There are people who have done much more, who basically started this field. And these studies are important. They were really met with skepticism early on. But what we learned from those studies, we learn both about fundamental characteristics of sleep, which are conserved across species and across phylogeny. And this is how we arrived to these basic defining characteristics of sleep. And we also learn about differences, about different ways how animals sleep and what does it tell us about sleep.
Are there some interesting kind of species-specific sleep patterns that you would like to talk about?
Yeah, that's an interesting question, in fact. So if we talk about mice, our laboratory mice that we study in the lab, we keep them in the 12-12 light-dark cycle, and we know that they're active during the night and they're mostly sleeping during the day. But this is in the lab. So if you take the very same mice and keep them in outdoor enclosure, they become diurnal or crepuscular. They actually can change their diurnality completely. And this may be influenced by many factors, such as light-dark cycle outside and other changes like temperature and food availability and the risk of predation. Sleep-wake pattern is much more flexible than we appreciate. So you probably wonder why mice in the lab, they're active during the night. Well, one possibility is that, of course, they do respond to environmental cues, such as changes in the light and dark, but also because usually during the day there are people around. So they don't like people, so they don't want to do their business when they're bothered all the time. So this is why they choose to sleep during the day and then engage in their wakeful activities during the night.
So this means that in nature, they're not actually nocturnal, or you said that's influenced by many factors.
It also depends how we define nocturnality and diurnality. So the classical approach is, of course, just look at when you are active. So if you're active during the night, you are nocturnal. When you are active during the day, you're diurnal. But visual neuroscientists would also talk about your visual acuity, your ability to see when light levels are very low. So nocturnality also refers to your enhanced ability to see at night, like the slow lorises that I mentioned. They have big eyes, so they actually can see in the dark.
So what are some of the factors that would influence the mice to stop being nocturnal? I mean, you talked about what the factors are in the lab, but if you put them outside, why should they then be active during the day?
Well, when you are outside, you have to kind of synchronize your behavior and physiology to the world outside, because it becomes much less predictable than in the lab. And this is primarily about the risk of predation. So you basically time your activity and rest with respect to the level of danger outside. Also, you take into account ambient temperature, because mice are warm-blooded, they're homeothermic, but they really need to spend a lot of energy to keep themselves warm. And they can also easily enter torpor, this state of reduced metabolism, to save energy. So they really have to weigh all the factors, like, so am I going outside now and searching for food, or should I better stay hungry for some time and save energy by becoming cold because it is too dangerous outside? These are extremely interesting decisions that the animals are making when they're out there in the wild, where you have your endogenous clock, your timekeeping system that counts your time awake and asleep, that we call sleep homeostasis. But you have to coordinate this with the unpredictability of the environment. And this is the real challenge. And this is how sleep evolved.
It's extremely interesting that they can change between diurnal and nocturnal. I really didn't know that. What I'm wondering is, when an animal is nocturnal, then it will have certain physiological rhythm in body temperature, in different hormones, in behaviors that is fixed with respect to the circadian rhythm. So does this mean that these things actually invert? So the peak in the body temperature will occur during the night in the lab, but during the day if they are outside? Or is it that their behavior goes out of sync with the physiology?
Well, I would say that definitely more research is needed to understand this phenomena better. Ideally, you would need to track the same individual animal, first in the lab condition and then outside, and then monitor all these parameters and then manipulate them in some way to understand the dynamics. So there is still very little known about how this happens. But when you mention circadian body temperature fluctuation, a lot of that is driven actually by you being awake and asleep, right? When you are active, your body and the brain are warmer. So a lot of what we call circadian fluctuations in temperature are actually, at least to some extent, accounted for by your activity and rest.
You mentioned several times torpor, and I wanted to ask you about torpor anyway. So I wonder if you could first explain what it is, and then maybe tell us about some of the work that you've done on torpor.
Torpor is an extremely interesting adaptation to harsh environments when it is cold and there is not enough food. Many animals, including some primates, have this capacity to enter the state of torpor that refers to the state of decreased metabolism. So you consume less glucose, less oxygen, your metabolic rate slows down. And this is an absolutely amazing adaptation. This is almost like a totally different dimension of existence. So we used to think about ourselves like we are either awake or asleep, and we exist within this very narrow range of metabolic regulation. But animals, including our close relatives, can step really outside of this comfort zone. And this is what many animals do when food is scarce, when you have to work hard to get the food. What you can do, you can enter state of torpor when you suspend your metabolism. You don't have to spend a lot of energy to maintain your body temperature. You save a lot of energy. So why I got interested in that, especially lately, is that there's a lot of confusion in the literature with respect to the relationship between torpor and sleep. So when animals are in torpor, they look like they are sleeping. So they assume sometimes sleeping posture, but there is a categorical confusion in a way. We define sleep and wake by behavior and brain activity, and we define torpor by changes in metabolism. So I was actually at some point surprised that we are still trying to compare this state like apples and oranges, while it is possible that you can be awake and asleep while being tortured. It is not something mutually exclusive. But very little is known about mechanisms that produce torpor and how this continuum between euthermic wake and sleep and hypothermic or hypometabolic wake and sleep, how they develop, how they are regulated. Very little is known about that, so I'm quite interested in this aspect.
So I know you've worked on torpor in hamsters, but do animals like rats and mice also enter torpor?
Hamsters enter what we call daily torpor. This can be triggered just by shortening the photoperiod, like in the winter. You don't even have to reduce ambient temperature. You just make the day short and they think it is winter, so they start losing weight, they turn white, and they start entering daily bouts of torpor when they spend several hours every day, basically a room temperature just above that. Other rodents can also enter torpor. Rats, as far as we know, somehow lost this capacity. We do not know why. Mice easily can go into torpor when they are hungry. So you can remove the food from their cage, and just after 24 hours, just one day, some of them will already enter torpor because they have to spend a lot of energy to stay alive. And some of them decide to not risk that and they start saving energy.
And roughly how much energy can they save? I mean, can you put a percentage on it?
You can kind of calculate if you decrease your metabolic rates to 30%. This definitely gives you a lot of flexibility. You can skip breakfast, lunch, and dinner.
And is this, what we might colloquially call hibernation, is it the same thing or is it something else?
Hibernation is a form of torpor. Torpor is an umbrella phenomenon that refers to a variety of different states when your metabolism goes down. And hibernation is a winter torpor. So it is a very deep, long, multi-day bouts of torpor when body temperature can reduce to 0 degrees or even sub-zero in some animals. So this is what we call hibernation.
And I've definitely read that in hibernation, some animals will actually warm up periodically in order to obtain sleep-like brain activity. Is that true?
Yes, you have kind of two questions in one. So I would definitely agree with the first one. At least in several species, it was shown that the animals warm up every few days, every few weeks. And this is quite amazing. They spend tremendous amount of energy to warm up. They reach euthermic state. And indeed, there is evidence that they spend a lot of time sleeping during that time. Whether this establishes causality, if they wake up to sleep, I think still remains to be established, but this is a very interesting hypothesis.
I suppose the reason it's interesting to me is because it suggests that whatever is happening in torpor, it's not giving them the type of processing that happens in sleep that is somehow necessary. I mean, there is this idea that sleep is necessary for health, And somehow, even when you're in torpor, you still need that type of activity. So what you're saying is maybe that's a bit of a stretch to say that it's a good hypothesis. We need to test it.
Yes, we need to test it, but you can also look at it slightly differently. Some of our colleagues consider sleep as a default state. So basically, you would sleep if there is nothing else to do. Imagine that maybe they just need to rewarm, to perform some cellular maintenance functions that require normal body temperature. It could be something very trivial, like they need to get rid of some toxins that accumulate in the body. And this can be done only when you're warm. And then what would you do if you're somewhere underground in the burrow, there is really kind of boring environment? You would probably sleep, and then some sort of mechanism would then trigger an initiation of the next torpor bout. This could be linked to some changes to the brain that is associated with hypothermia. So I find this idea quite interesting.
I find it quite interesting as well. And I know you have a paper where you looked at how sleep after torpor differs from sleep after sleep deprivation. After torpor, there's an increase in slow waves, and there are steeper slopes in the slow waves, which you would not find after a night of or a period of sleep, I guess.
Yes. So animals go into deep sleep, if we define depth by the occurrence of slow waves, after both sleep deprivation and torpor. This is really a striking phenomenon. So if you look at this recording showing a profile of slow wave activity around a torpor bout in a hamster, you see this absolutely remarkable increase in slow activity right after the animal emerged from torpor. So it looks precisely like sleep deprivation. So what we found is that slow waves after torpor seem to be slower, so their slopes are more shallow. What this means, we do not know. But since there was this link established between slow wave slopes and the synchronization in cortical networks, one can conclude that torpor has different effects on the network connectivity as compared to sleep deprivation.
And can you speculate that torpor at least doesn't lead to such a deep sleep need as sleep deprivation? I mean, given these shallower slopes, it would suggest that the drive for sleep is not as strong.
The truth is we don't really know what slow wave activity after torpor or sleep means functionally. It is possible that it is somehow linked to synaptic function, because we know that during hypothermia or during torpor there are some changes in synaptic connectivity associated just with hypothermia. We also know or suspect that something of that kind may be happening during sleep deprivation as well. In this respect, torpor is a very interesting model for this occurrence of natural synaptic plasticity, because this is such a peculiar state when not only the body but also the brain goes into something completely different. The remarkable thing, the animal comes out of torpor and it is still the same animal with same memories, so it still remembers its neighbors or where to find the food. So there were a few studies suggesting that memories are actually retained, or most memories retained after emergence from torpor, despite such a massive stress.
But do we know whether there's any consolidation of memories? Has anyone looked at whether memories are strengthened or integrated somehow during torpor?
Well, very little is known about that. I would be surprised because it is a state of very much reduced metabolism. There is not much neuronal activity. Neuronal spiking is very much reduced. As far as I know, no one looked at the capacity for, let's say, LTP induction during torpor. That would be very interesting too.
But what about whether synapses are strengthened or weakened across torpor? So if you have some measure of the strength of a synapse before and after, does it change?
According to the literature, torpor is associated with a reduction in synaptic connectivity. So how is it restored in these first few hours of euthermia? What are the factors? Do you just need to be warm for that to happen, or do you need to be asleep? This is one of the questions on our agenda that we want to study.
Mm-hmm. But when it's restored to the same level of strength as before, or you don't know?
According to the existing literature, yes, it seems it just comes back, but the time scale is, of course, unknown because most of the studies which you can really trust are anatomical studies, and then, you just have one time point per animal. So we need better and refined techniques to monitor individual synaptic connections across the torpor bound.
Oh, it's fascinating. Okay, so I wanted to ask you about local sleep. This is something that you have some very nice publications on. Can you maybe explain what it is and talk about some of the work you've done?
Traditionally, initially, sleep was defined as a behavior. So when the animal or human assumes a specific posture, you become partially disconnected from the environment, you become less responsive anyways. So we define sleep as a behavioral state. And behavior, the organism is less likely to be asleep and awake at the same time. So you would be awake or asleep. Clinicians, of course, saw some interesting cases of sleep disorders where you can satisfy criteria of wake and sleep at the same time.
So even behaviorally, that suggested that you could have some kind of a partial sleep or local sleep in a sense. I see what you mean.
Yeah, but if you look closely and carefully at this traditional defining characteristic of sleep, none of them is sufficient or necessary. You can be perfectly awake while being immobile, and this is what makes it challenging to use the criteria that we developed for some animals and then to apply them to other animals that may sleep in different ways.
So that's the kind of behavioral level. But was there any clinical data that started to suggest a more intracranial level partial sleep, or was it all behavioral?
So here probably I wanted to mention a couple of other observations that really stimulated this area. So one is the ideas of James Kruger, who started talking about local activity-dependent or use-dependent aspect of sleep. He was one of the first who suggested this very explicitly together with Ferenc Obal. So they wrote several quite influential reviews on these topics, explicitly stating that sleep occurs at the local level as a result of or in relation to activity. They did draw evidence from other studies. One famous study was conducted by Ivan Pigarev, who is a visual neuroscientist. So he was not interested in sleep, but he accidentally noticed in one of his experiments that when a monkey that was performing a task while he was monitoring neuronal activity would continue performing the task as it were awake, but some neurons stop firing. And he thought that this is how local sleep may start at the level of individual neurons. Then the studies by Mukhametov and Lamin on sleep in dolphins and marine mammals were, of course, hugely influential here, which really make this case very strongly that, first of all, you can show behavioral wakefulness, like even movement, while at least partially you are sleeping, and just to show this absolutely dramatic example of lateralized sleep. And then I want to mention the observation of burst silence pattern of neuronal networks that were first published, again, very long time ago, in like 1930s, that if you culture a spinal cord or some piece of nervous tissue, it can enter the sleep-like mode of activity. It was most carefully investigated by Michael Corner, who considered this type of activity of burst suppression as a primordial, primitive type of activity, and this probably can tell us something about how sleep evolved. This type of activity is considered as a primitive type of sleep or sleep-like activity.
And isn't it almost shown to be a kind of a default activity for some in vitro preparations? I guess if you remove the acetylcholine, then they go into this oscillating state, don't they?
Yeah, well, you don't have to remove, you just don't add any wake-promoting neuromodulators. And then as more recent studies also from Mehdi Tafti's lab show quite beautifully how neurons in a culture, after some days, they establish connections and they enter this state of burst suppression, which looks like sleep, or I would always, when Mehdi presents it at a meeting and he calls it sleep in a dish, I ask him, but maybe it is coma in a dish. So we are still kind of debating what exactly is that, but it is very similar to what the brain is producing normally in the brain when the animal is sleeping. So there is this activity.
One thing I wanted to comment on in what you just said is the part about the kind of split brain sleep in marine mammals. So this is the observation that they can sleep with one hemisphere at a time, which is clearly a sort of a simplified version of local sleep. And what I wanted to comment, I just can't resist. So there's been a recent work on seals showing that they can actually change this property depending on whether they're in their aquatic phase or whether they're in the phase where they live on the land. So if they are aquatic and they're spending all their time in the water, then they use split brain sleep, and that allows them to sleep with one hemisphere and keep swimming and obviously keep breathing. But if they're living on the land, they shift to regular whole brain sleep, and they actually then introduce REM as well. So they don't express REM while they're swimming, probably because REM requires paralysis of all the bodily muscles. So they wouldn't be able to continue swimming if they're in REM sleep. I don't know if you can have split brain REM sleep. Is that something that has been looked at?
Yes, this observation that seals can switch their sleep pattern depending on water and land tells us two things. One is that bihemispheric sleep is probably still preferential, because if you develop the circuitry to produce unihemispheric sleep, why would you go into bihemispheric when you don't need it, right? So this means that it has some advantages. But on the other hand, it of course tells us that sleep is still very important, because you rather develop unihemispheric sleep in water rather than just get rid of it altogether. It tells us also something important about flexibility of sleep. And this is really quite striking, that animals, depending on the conditions, on environmental demands, they can make such dramatic change. But this, of course, also tells us something about our own condition. It seems we can do so little about our sleep. And you almost get envious that these animals can change their sleep pattern completely, right, without any effort, apparently. And we go to bed in the evening and we even struggle to fall asleep.
Well, maybe this is something we can work on in future with kind of sleep engineering and design of sleep and all of the strategies and technology that are out there. But I think it's going to take a while before we are as good as seals.
Yes. But going back to REM sleep, about REM sleep being partial, this is an interesting question. It is also a difficult question, because REM sleep has not only brain-related phenomena, but also peripheral phenomena, such as muscle atonia. I would say REM sleep is more like state of the organism rather than the state of the brain only. Talking about brain activity, of course, I have no doubt that there can be partial REM sleep. We know the literature is accumulating that you may have slow waves. You may have some areas of the brain that basically never go into activated pattern. So they always stay in a default non-REM-like mode. Again, neither non-REM nor REM sleep is a homogeneous, uniform state, but there is a lot of complexity.
Yeah, I agree that there's a lot of complexity. So let's go to your paper with Yuval Nir. Can you just maybe talk a bit about what you showed there?
This was based on data collected in patients with drug-resistant epilepsy who were implanted with electrodes in various parts of the cortex and the hippocampus and amygdala, I believe, to find the origin of seizures. These patients, they are very happy to participate in experiments because there is not much to do otherwise. And this is how Yuval collected whole night recordings in these patients. And they had this very rich data set with recordings, intracranial, both multi-unit activity and LFPs in the same patients. They found that slow waves, which is one of the defining characteristics of non-REM sleep, they basically never happen simultaneously everywhere. So if you look really at the local level, you can easily find parts of the brain that stay awake, they're activated, and then a few centimeters to the right, another electrode shows clear-cut slow wave. This was the basic observation of that study.
Yeah, and then, some years later, there was a paper from Gina Poe showing that different brain areas could be in different stages. I think the hippocampus and areas of the cortex could be in REM and non-REM at the same time, basically. And the hippocampus was often leading into REM, I think. And there was kind of a complex pattern going on there.
Yeah, this is a nice example of when you look carefully at the data and look at kind of from a different angle at something that you take for granted. And this is how you make such very interesting and important observations. Because we know that the animal doesn't go directly from non-REM to REM sleep. It goes what Claude Gottesman described as an intermediate stage with spindling, and the hippocampus was already in theta, and Gina looked at it very closely. And it's really quite striking that some REM-related phenomena may coexist with non-REM-related phenomena elsewhere. So this entire subdivision of sleep into two states is a big mystery to me, to be honest.
I think it's a really interesting mystery. And actually, I talked to Gina about this in a different podcast. So people listening might have heard a little bit of that story. But I think then your paper where you looked at local sleep in the more sleep-deprived situation actually kind of shed some light on that mystery, because now you're looking at maybe the brain as it's falling asleep or parts of it are falling asleep, instead of Gina was really looking at the brain within a night of sleep, right? So it's slightly different situations. So can you tell a bit about that paper?
Yes. In that study, we recorded from a few cortical locations in rats who were sleep deprived. What we did, we just encouraged the animals to stay awake by giving them novel objects to explore. And they somehow accumulate sleep pressure. And what we noticed is that the longer they stay awake, the more likely they would produce very clear-cut slow waves occurring in just one local area of the neocortex. The longer the animals stay awake, the more likely they would enter just for a very brief moment of time, like a local sleep state.
So these were rats who were performing a task. They were sleep deprived and they were performing a task where they had to reach and get a sugar pill, which they like to eat, obviously. I can't remember if they had to press a lever or something. And you were recording in the motor cortex, is that right?
Yes, that's right. The way this experiment worked is that actually first, before even implanting the animals, I gave them this opportunity to reach for a sugar pellet that they like. Normally they're getting just a standard rat food. And then imagine when you're given something much more tasty. It was, I think, banana smell sugar pellet. And of course you want it, right? But if it is slightly outside of your reach, so you cannot reach it with a snout, that all of them tried. At some point they realized that the only way to get the sugar pellets is to reach with a paw. So this is an extremely unusual situation for the rat, right? So they never had to do anything of this kind in their life. So first they have to decide if they're left-handed or right-handed. And very quickly they would make up their mind. So it was really not random. Some of them would start using the right hand, another left hand. So the split was roughly similar to what we have in humans. And then they would use one paw consistently. And so I would take them and implant the electrode in the motor cortex and the side contralateral to the dominant paw, because I knew that they will use that particular paw. And this gave me the opportunity to see what the motor cortex is doing exactly in the area that controls the movement, especially the learning aspect of that particular paw.
I see. So you had to watch them doing the task, figure out if they were right or left-handed, and then implant the electrode appropriately contralateral to the hand they're going to use. And then you could record the activity associated with that task.
Have you seen a newborn rat?
No.
It's like a tiny red or pinkish color creature, completely blind, completely naked. But already at that stage, it is possible to predict if it will be right-handed or left-handed. The way you do it, you take this almost like an embryo, you put it on the flat surface, and you see in which direction its tail curls. So it is never straight. It goes either to the right or to the left. And this would predict if they become left or right-handed. They have this intrinsic laterality. Well, yeah, I did not do it in these studies. We got adult rats, so we had to figure it out experimentally.
So you recorded from the appropriate contralateral cortex to what they were using. And those were the neurons. So they're actually motor cortex pyramidal neurons that showed this pattern of kind of pausing in their firing when the rat was sleep deprived.
The technique we are using, the extracellular recording of multi-unit activity, does not really allow to know much about the identity of those neurons, but those neurons were in the area which is supposed to be important for executing this reaching movement. This was established in other researchers who would place a lesion on that particular area, and then the rats would completely forget the task, so they could no longer execute it.
And so from memory, what you found is the more sleep deprived they were, the worse they got in the task. And also the more you found that these neurons showed basically slow oscillation down states. So they paused in their firing. So they had the equivalent of a down state. It looked as if they were doing slow waves, basically.
Yeah, it was slightly different. So we did not look at how their performance deteriorated as a function of time awake. What we found is there was an association between a miss and the occurrence of a brief neuronal silence.
But you didn't look in other areas, so you don't know if this...
We recorded also another brain area, and there we could not find the same association.
So would you speculate that when we are sleep deprived and we are starting to lose concentration, for instance, other areas of the brain are probably showing this pattern?
First of all, I'm pretty sure that a lot of our brain cells are not engaged at any given moment in any case. Many of our neurons are probably idling one way or another, and there is indeed evidence for that. But I think indeed if neurons are not engaged in the right kind of combination with other neurons at a particular moment of time, I can easily imagine that this may result in some behavioral deficits.
Yeah, so this is another thing that would be quite interesting to examine more and maybe to examine it in humans as well.
Yeah, there is a certain point when your performance is optimal, but then if there is too much activity or too little activity, this will compromise your performance. So I think it is not like a very simple relationship between neuronal down states and behavioral lapses. But you can also imagine that if the brain enters a hyperexcited state, this may also compromise your performance. But yes, indeed, this study suggested that if you are sleep deprived, if you are tired and the brain starts entering sleep-like state at a local level, this may result both in our subjective feeling of being tired and also in objectively measured deficits in performance.
But of course, it's not only when we're tired, right? So you've also looked at this in rodents who are running on their running wheels obsessively, addictively, and a lot, basically.
That's right. It is possible that in the absence of activation or stimulation, neurons may enter a silent state. So it was shown for several other brain areas. I remember for the auditory cortex that in the absence of auditory stimulation, even an awake animal, auditory cortex may show sleep-like activity. And in our study, we showed that definitely even in a highly behaviorally active animal, neurons may stop firing altogether. You may see sleep-like slow waves. So I think really we have still insufficient understanding of this continuum of states, both behavioral and brain states recorded electrophysiologically, and you can imagine any possible combination of this, right? So you may be highly active and show sleep-like activity, and you can show a lot of activity in a very deep sleep.
But just to be really clear about what you showed in that study, so you had rats who were running on mice.
Yeah.
They were running on running wheels. And when they were really getting into this, you found in the same motor cortex that you had been looking at before, the same kind of pattern of neurons showing temporary silent periods for several milliseconds.
Or even more than so. We saw neurons which completely stopped firing when animals engaged in a very stereotypic, simple running pattern at a high speed. And this is when, on average, there was a reduction of neuronal activity in the motor cortex and the somatosensory cortex. But some neurons, they would just completely stop firing in the motor cortex, in the very area that is supposed to be related to movement control. And we speculated that this happened because you effectively do not need the neocortex when you're engaged in very simple behaviors. Like when you go jogging, for example. So some people report this interesting state of mind wandering, which can have some similarities with sleep state.
So do you think of this pausing behavior as kind of a proxy to slow wave sleep, or you think of it as something else? Because if the cells are silent for a really long period, then it's nothing like a slow oscillation, right?
Yeah, it's different. Again, look at this as a continuum. So if you go from very superficial sleep to a comatose state when neurons are effectively silent for long periods of time, and then, yes, if you look at this behavior of this neuron without knowing what the animal is doing, you can say, well, this is really a comatose brain.
Okay, so comatose rather than sleeping. That's the distinction. Yeah.
Yeah, well, prolonged periods of complete silence, I wouldn't call it the same as down states during sleep.
So how long?
I think we have in the paper a supplementary video that shows some quite remarkable examples. When basically neuron was active only when the animal slows down or speeds up, and otherwise it was totally silent.
It makes me wonder what's happening in my brain when I'm jogging every day during lockdown at the moment. Okay, so now I have to ask you about sleep in plants. I know the paper is about learning in plants, but I believe there was also a sleep element to that. But yeah, you can tell me.
Okay, that was an interesting collaboration that in fact started with the question whether plants can sleep. Plants, as you know, have circadian clock, and the first evidence of circadian clock was actually made in plants. So you probably know the story of mimosa.
So I actually just read this story again yesterday as it happens. So I think it was that these mimosas, they open up the leaves during the day.
Yeah, exactly. Yes,
and then they close them during the night. And they have a strong circadian rhythm, which was thought to be entrained by light. Then he put them in a dark sealed box. And then occasionally he kind of peeked, but in dark conditions still. And he found they are still exhibiting exactly the same circadian rhythm, even when they have no access to light at all.
Yeah, so which suggests that these movements are not imposed from outside, but the light-dark cycle, but they are endogenously generated. So definitely plants have the circadian clock. If you look carefully at the biochemistry, they can share a lot of similar pathways and receptor subtypes with animals, including some subunits of glutamatergic receptors. There are studies that show that you can anesthetize the plant. So if you take the same plant that shows movement and you put it in the chamber with isoflurane, they stop moving. Yeah, so the question is, do they sleep? And the way it happened is that there was a New Yorker article about Monica Galliano, who is the plant researcher. So she started as a zoologist, as an ecologist. And at some point, she decided that she does not want to work with animals for ethical reasons. And I got in touch with her. I wrote her an email and asked her what she thinks of plants sleep. And she was very excited to establish the contact. And we had several meetings discussing how an experiment can be designed to look at sleep in plants. It was just not straightforward because you would have to use the criteria developed for animals and somehow enforce those criteria for defining sleep to the plant. But she was very keen to explore the capacity of plants for learning, and she already had a few studies showing that plants are able to habituate to repeated stimuli. And then she designed this experiment about their learning capacity, where she would pair the light delivery with a neutral stimulus. And it turned out that the plants actually care about that. So she would compare it always with Pavlov's dogs. So this is how they learn the association.
So my memory of this is you paired the light with a fan that was blowing air. So the plants turned towards the light because that's what they do. And eventually you took away the light element, but you maintained the fan and you found that the plants turned towards the fan. So they had learned that association.
Yeah, so basically the fan stimulus always preceded light delivery. So plants have this innate phototropism, so they would always grow towards the last presentation of light. And you could override this natural drive by the presentation of fan. So they could start growing in the direction of fan, even though light was last time seen on the other side, because they learned from before that fan always predicts light.
Oh, so it wasn't that they turned across 24 hours. It was that they grew in that direction.
They grew, yeah.
So it's still much more than people typically think plants can learn, right?
They can really do amazing things.
So was there a sleep element in the experiment as well, or?
There was a circadian element because we tested this capacity to learn at different times of day, and only during the day when light is normally there and it is meaningful, they learn the association. But during the night, they are less responsive, and this makes a lot of sense. For plants, light is not just the stimulus that signals the arrival of the day, like for us. It is really their food. Light is everything for them. So the entire physiology is completely dependent on the light. But circadian aspect is hugely important for many things.
Is there some other topic that I really should be asking you about that you're excited about at the moment, or that you think is particularly exciting?
You mentioned that I'm studying what sleep is, and maybe I just want to bring this up again as a very important issue. We always kind of take a shortcut and then we want to find out what sleep is for. And there are two aspects of why we sleep. It is like how come and what for. And I don't think we can get to what for without understanding better how come. In my lab, we are taking many different directions because I think sleep is just by definition such a phenomenon that you cannot possibly understand if you look at just one aspect. This is why we look at metabolism and circadian rhythms and neocortex and the hypothalamus and different species.
So you're taking a much more holistic approach and you're really trying to understand what it is as a phenomena.
Some people dislike the term holistic, but I think it's a good way to put it, yes.
I think this is a really good place to stop. But thank you so much, Vlad. It's really interesting to hear about all these very disparate examples and ideas in different species and paradigms about sleep.
Yeah, thank you. It was a pleasure talking to you.
You've been listening to the Sleep Science Podcast with me, Penny Lewis. My guest today was Vlad Vyazovskiy of Oxford University. The Sleep Science Podcast is a production of the NAPS Sleep Lab at Cardiff University, and our producer today was Dominic Carr. If you've enjoyed the show, please consider subscribing to the podcast or liking us on Twitter. If you have questions or comments, please get in touch via sleepsciencepodcast, all one word, @gmail.com. Thanks for listening, and until next time, sleep well.